So this mini lecture is to introduce you to some of the key scientific experiments that led us to understand the structure of DNA and a little bit about how it's replicated. So the only one missing from here is in 1958. The last slide we'll talk about is Meselson and I don't even know if I'm spelling his name right. No, let's try this again. Meselson and Stahl, I knew there was an H there somewhere, who showed that DNA replication is semi-conservative. So you don't have to know. Everybody on here, take a look at the Chapter 10 objectives. We're going to talk about measure. Let me see if I can see all these. Where's Griffith? Where's Chargaff in here? Avery, MacLeod, McCarty, Hershey, Chase. Oh, there's Chargaff, Watson, Crick. Oh, it doesn't have Wilkins and Franklin. Frankel and Conrad. And Meselson and Stahl. So just kind of a timeline, and what I want you to see really is how much happened in about 10 years from figuring out that DNA was the genetic material to its structure and starting to understand how it replicates. So I love this guy just because who wouldn't like someone who studies pus from Band-Aids? So Meisher, and I apologize if I'm saying names wrong, studied the pus, really the white blood cells, which are these big cells with a purple nuclei, from wounded soldiers. And he went around collecting these pus-filled Band-Aids and started isolating the nuclei, these big purple blobs, from white blood cells. So, in case you don't know, red blood cells do not have nuclei, and the white blood cells, you can see, are much larger. So he was able to isolate these nuclei and determine with some chemistry that they were slightly acidic, or the chemistry of them was, and it contained a lot of phosphorus. So this was our first insight into that there's something in the cells that have this phosphorus and this acidic characteristics. And he called this nuclein. His apparently graduate students renamed it nucleic acids, and that's where we get that terminology for DNA and eventually RNA. But they knew in the nuclei there was both this substance and there were proteins. And really what was going on for the next 50, 60 years is trying to determine, was it proteins or was it nucleic acid that was the genetic material? And because proteins had 20 amino acids and seemed so much more diverse than the 4 nucleotides in DNA, most scientists figured protein had to be the genetic material. So we're going to talk about the evidence that brought us to figure out that DNA actually was the genetic material. So years later, like I said, in the 40s and 50s, when this all kind of exploded, Chargaff analyzed some DNA. So we knew it was deoxyribonucleic acid, just didn't know exactly what it was doing. He isolated DNA from different organisms, and he analyzed the base components. So if you read the chapter, it talks about some of the people who first figured out that there were 4 bases, and that DNA bases were made of a sugar, a phosphate, and a nitrogen base. The big important contribution from Chargaff was that he found these ratios. And he found that the amount of A equaled the amount of T, pretty much. And the amount of G equaled the amount of C. But the amount of A did not equal G, and T did not equal C, and so forth. So there was something interesting happening with A and T being equal and G and C being equal. And this is eventually going to lead us to the base pairing rules. The other thing he noticed is that different organisms had different amounts of the different bases. So there was not a standard level, right? So here E. coli has 25% G's and C's, whereas yeast only has 17 or 18%, and humans have 20%. But when you did the ratios, no matter what organism they came from, the amount of A and T were equal and G and C were equal. And the other important thing is you can look at the ratio of A and G, which hopefully you know are the purines, is equal to the ratio of T and C, which are the pyrimidines. So again, putting this together later as Watson and Crick determined the molecular structure of DNA, this was a key bit of information to help them put that model together. So people were studying chemically, biochemically DNA. Other people were trying to figure out, is it proteins or is it DNA that is the genetic material? And a really early experiment by Griffith in 1928 came up with this idea of that there is some kind of transforming factor in a cell that is heritable. So let me walk you through his experiments. So Griffith worked with bacteria, and this particular bacteria was Streptococcus pneumoniae. And he had 2 strains. He had a smooth strain, which he called type 3S for smooth, and this was a virulent strain, which means it causes disease. And he had a second strain, which was called a rough strain, or 2R, and this was avirulent, which means did not cause disease. So he did some work in mice, and he took the smooth virulent bacteria and he injected into a mouse, and the mouse died, got disease. And, whoops, sorry, when you isolate or culture from this dead mouse, you got back the virulent bacteria. So it must have replicated, it was still viable, and it killed the mouse. This was in contrast to the rough, the avirulent type 2. When you injected that into the mice lived, and you couldn't recover any of that type of bacteria, indicating, which we now know, is that the immune system was able to overcome this bacterial infection. So you have a virulent strain, you have an avirulent strain. What he did next was he took the virulent, smooth strain and heat killed it. So he basically heated up the bacteria, killed it, put it into the mouse lived, no bacteria recovered. So this was kind of a control experiment, because the next was the most important. The next experiment, he took the heat-killed virulent strain plus the rough strain. Okay, so alone, these two mice live. He took the heat-killed strain, he mixed it with the avirulent strain, injected it into the mouse, and the mouse died. So all of a sudden, somehow, the avirulent strain was transformed into a virulent strain. Right? Somehow this combination killed the mice, when alone they didn't. And when he went to isolate bacteria, he got back the smooth strain. He got back the virulent, even though it was heat-killed. So what Griffith discovered was that there was some factor, something in the cells, that could transform a non-virulent strain to a virulent strain with the same characteristics, right? Back to the smooth. So it was the rough that was alive, and we got the smooth back. So something transformed it. And that something was likely the genetic material. But it wasn't till about 20 years later that we could figure out what that transforming factor was. So Griffith did not figure out it was DNA or anything like that. He just said there was some transforming factor. Avery, MacLeod, and McCarty took his experiment and went a step further. And so what they did was they took this virulent bacteria, same stuff Griffith was working with, heat killed it. Homogenize means like put it in a blender, right? Blend it up. Filter it. So they got rid of lipids and carbs. They got all that gooey stuff out. And so they had the components from this bacterial heat-killed virulent strain. And one by one, they eliminated different macromolecules. And we'll walk through this in a minute. But what I want you to see is they eliminated a macromolecule. They mixed it with the rough strain, so they're repeating Griffith's experiments, injected it into mice, and looked to see what happens. Okay, so when you added an enzyme that destroys RNA and you mixed it with bacteria, you still got the virulent back. And if you put this into the mouse was dead. Okay. So the transforming factor was still working in this experiment. In the second experiment, they added proteases, which are enzymes that destroy proteins, mixed that with the rough bacteria, and by goodness, they got the smooth back, the virulent back, and if you injected this into a mouse, killed the mouse. So the transforming material was still there. So finally, and who knows if it was even in this order, probably not, they took an enzyme called DNase, which is an enzyme that chops up DNA, added that concoction to the rough strain, and only got rough back. No smooth. The mouse is happy. No transformation. So their conclusion was that when you mix the destroyed DNA with the bacteria, you get no transformation. Therefore, DNA must be the transforming factor. Okay? So they figured out DNA was the transforming factor. On our way to thinking that DNA might be the genetic material. A few years later, Hershey and Chase, and that shouldn't be Hershey Chase, it should be Hershey and Chase, we're looking at phage. So bacteriophage are viruses that infect bacteria. And a phage is basically made of a protein coat, or in this case head, and DNA inside. And so they started using what we call radioactive isotopes. This is a way you could follow things or measure things before we had all the molecular biology tools we do now. So for one group of phage, they grew them in 35S, so radioactive sulfur, which made the proteins, shown here in yellow, radioactive. So the way a phage works is it binds to a cell and it injects its genetic material into the cell, and more phages are made. So the question was, is it injecting the DNA or is it the protein that's going in to make the new phage? So they radio-labeled the proteins, they allowed an infection to happen, they put it in a blender, which you've got to love that technology, and all of the phage heads, they didn't know at the time, came off. Centrifuge, which means you separate out things based by weight. So all the cell parts were at the bottom. The liquid was everything outside of the cell, and they found out that the liquid was radioactive. So the protein did not go into the cell. Okay, so let's try the flip. They take P32, which is radioactive phosphorus. And you know that DNA has a phosphate sugar backbone. So they labeled the DNA radioactively, allowed the infection to happen, blended it up, separated it out, and now in the cells was the radioactive material. So that meant that the phage were injecting their DNA into the cells, not the protein, and that was making new phage. So it had the information, to make new phage. And so that for viruses, DNA was the genetic material as well. So we have evidence that bacteria are transformed by DNA, genetic material. Phage inject their DNA into cells to make more phage, genetic material. So everyone started focusing on DNA. Also happening at the same time was Franklin and Wilkins, who were looking at the structure of DNA. So they used a technique called X-ray diffraction. It makes an image, and they could determine from this that DNA was in a helical form. So they didn't know the structure yet, but this information gave them some size information. It said that this was a helical molecule, and from that, Watson and Crick took this information and put together, with kind of a Tinker Toy set, the 3D structure of DNA. So they had all this information about base pairing and a helical structure. And they figured out that the strands had to be anti-parallel. And they figured out the purine-pyrimidine base pairing. And probably what was most convincing of their structure is that it would lend itself to replication. Right? And this is really important because we know genetic material not only has to be passed down, but it has to be replicated from cell to cell. DNA isn't the end of the story. And if you ever get a chance, there is an RNA world hypothesis that we think that RNA actually was the first genetic material. RNAs can be catalytic, they can self-replicate, and they are the genetic material for many viruses. So this is tobacco mosaic virus. And it is made up of RNA with a protein coat. So very similar to phage, but with only RNA. And Frankel, Conrad, and Singer did some cool experiments where they took different strains of TMV virus, they removed the protein coat, and kind of exchanged it. So now they had protein, no, here it says right here, RNA A covered in protein B, RNA B covered in protein A. So they switched the coats on the RNA molecules. And it's fairly easy to do because viruses will actually self-assemble in a cell. So if you give it the RNA or DNA and the protein coat, it'll actually self-assemble into a virus. They could infect this into tobacco leaves, and when they analyzed what kind of virus was made, they found that the RNA dictated the type of virus. Oops. Or the type of virus offspring. Oh, I'm really having issues. Wow. Offspring, sorry. Okay, so the progeny. So even though they coated this RNA A with protein B, didn't matter. The RNA A directed the production of protein A. And likewise, the RNA B directed production of protein B. Okay. So this landed the idea that RNA was genetic material as well. And Greer and Schramm contributed to us understanding that RNA alone is infectious. So it's pretty cool. You can isolate RNA, you can isolate RNA, rub it on a tobacco leaf, and virus will be made. So RNA is important genetic material for viruses as well. The last experiment I want you to understand, which also uses isotopes, is Meselson and Stahl. And Meselson and Stahl looked at how DNA is replicated. And so there was lots of ideas. We understood the DNA double-strand structure, the helix, but how were new strands made? And there were three ideas. It could be conservative, where new strand stays with new strand. Okay, let's make this a little more clear. Old strand stays with old strand, and then you have a new one. Semi-conservative, where old is split. Or dispersive, where it's kind of this random, what do I want to say? Random back and forth between old and new. Random combination. Okay. So what they did is they had nitrogen, and N15 is what we call heavy, has an extra neutron, and N14 is the normal or light. And so they first made the DNA in the heavy nitrogen, and then let it undergo replication. And what they observed was that the DNA was a mix of heavy and light. So that meant that you could either have semi-conservative or dispersive DNA replication. If you had conservative, you would have had a band of light DNA and a band of heavy DNA. Oh, and I should say, this replication happened in the presence of the light nitrogen, so that you're switching isotopes, which allows you to follow these molecules. Okay, so one round of replication couldn't distinguish between semi-conservative and dispersive, so they did a second round of replication, and what they found was that some of the DNA was now only light, and some of the DNA was still a mix of heavy and light. Oops. And that corresponded with semi-conservative. Because if it was dispersive, you would still always have this random combination of old and new. But with semi-conservative, what you had is when your blue strand and yellow strand, let's see if I can do this, your blue strand and your yellow strand were replicated, right? All the DNA, new DNA is going to be light, so you would get a second light strand here and a light strand here, and that would give you light and a mix of DNA. So, take some time, read through your textbooks, look through this, and understand, be able to explain what each of these scientists discovered and the experiments that they used to figure out and contribute to our understanding of DNA as the genetic material. All right, thanks.